So I'm Mark from SolveBio, easily impressed by technology. So first of all, thanks so much for having me. This is it's a really great event. First of all, a lot of people, and really combines all my interests: health, data, people listening to me talk. So this is really awesome, and the other presenters have just been amazing. So hopefully I won't be boring. So first off, a few words about what we do to give some context. So we want to be the data infrastructure behind genomics. So what that means is we want to use data to help interpret the genome for clinical and health purposes. So if you go to the doctor and go to Recombine and get your DNA sequenced, it usually involves ingesting a lot of data from the outside world about what different DNA sequences mean. And we help provide that data. We normalize it, put it in the right format, quality control, et cetera. And we think that that's really important since genomics is growing quickly. So I want to talk today a bit of a perspective on kind of a new buzzword that's going around called precision medicine, and where I think genomics fits in, and where data scientists fit in, and how I think together that those communities can really make a big difference. Precision medicine is, the idea behind it is that medicine is becoming more and more data-driven. It's no longer supposed to be about guessing, more about you have a bunch of data, so now there's evidence that can be used, et cetera. So people are investing a lot in developing technologies to measure a lot of information about people. The problem is, the big concern is, what if precision medicine actually just becomes, we have a lot of data, but we don't know what to do with it yet, medicine. So why measure all this data? Why sequence your genome? And literally, why sequence your genome? I mean, for most people, there's no information that they glean yet from it. And you know, the big fear is that, kind of looking back 10 years from now, that precision medicine will look a bit more like the past than the future. That's actually my doctor, so don't laugh. And you know, the question is, what exactly can we do with all this data that's being collected? How does it actually translate to something that's, you know, medically actionable? And right now, we're not exactly quite there yet. So there are some definitely bad news in the headlines. First, missteps about using genetics. A lot of people are scared about it. So you know, there's a lot of really promising new technology. Alex gave a really great background on the field of genetics. This is an Illumina machine. It looks really, really fancy. This thing can sequence your genome for $1,000. And 10 years ago, it cost like $3 billion and was really hard. Now you just get one of these machines, kind of looks like an Apple, I think. It looks really awesome, and people are buying lots of them. Columbia has a bunch of them. Harvard, other prestigious names have them. Illumina is doing really well. They're worth $30 billion. They make these sequencing machines because people are just super excited about using genomics for stuff. And that's really cool. So you would think that, and also, okay, there's also this thing, wearables. This can tell you if you have a pulse or not, how many steps you've taken, and whether you're sleeping. So, and then there's this thing just launched. Looks really, really cool. I don't know what it exactly does yet, but it's really awesome. Also looks like Apple. I think we're getting somewhere. So this is awesome. So you would think that this is what the doctor's office looks like. This is your doctor now. It's what I assume is a Bloomberg terminal. And there's all this data coming in real time. I know everything about you. All this, your DNA, your sleep schedule, all this whatever fitness stuff that you do, and your doctor's there, he's eating lunch, and he is deciding what to do with you. That's really awesome, and that's, oh my God, this is actually what it looks like right now. And this is literally what the data behind genomics, behind precision medicine looks like right now. FTP sites, I know everyone just got sad looking at it. This is my life. This is a lot of genetic. So every genetics company, every hospital, they design and implement tests to figure out what disease you have or don't have, or whether you should have a child, or what drug to take based on your DNA. They use this data that's collected by academics and deposited in a folder called data somewhere on FTP sites. And this looks like the 80s, but this is actually from like last week. That's true. This is true. So 23andMe uses this data and papers. Council, Recommize, I don't want to speak for you, but everyone uses this data. And it's valuable data. This is data that academics have collected over the years. And it's really great. And there are even instructions for how to use it. It's a README file. So it's great data for the first kind of chapter 1 of genetics, but the problem is that there's no quality control. You don't know which records are right, which records aren't relevant. There's no feedback for this worked for me, this didn't work for me. There's no system of knowing which version you used, which you didn't use. And this, if people in the industry are worried about regulatory backlash or consumers being underserved, this is definitely one of the pain points, and it's scary. And frankly, it's really scary. There are some great companies in this space that are doing a great job on the software end, as Alex mentioned, but then there are other companies that might not be data scientists. They might not have software. I'm really scared to go to those hospitals or those companies and know how they're using the data from the outside world to determine what disease you have or what you should do. By the way, where's the time thing? I know it's somewhere. It should be looking somewhere. Oh, okay. Just let me know if, okay. Okay, so so the main kind of thing that we're interested in at SolveBio, and I think as a community we're interested in, is we've been able to generate as a community a lot of value from knowing a very small amount of information. What DNA variants, the things that make us all different, which ones cause common diseases. And there's still a long way to go to being good at measuring those. And there will be multi-billion-dollar companies that will figure out how to measure those efficiently, deliver that information to the patient, and get feedback from the patient. There's a lot to do there. But the interesting thing is that that's just the tip of the iceberg, the tip of the iceberg. It's crazy. There's so much information that we don't know what it means for your health or for medicine. And if we can collect and understand that information, so over the next few years, we're just the genetics world, because there's so much capacity, they're collecting so much information, they're observing so many new DNA variants, and none of them are being understood or explained, or very few of them are. That's a huge opportunity. If people can understand that, then they could truly transform health. And I want to tell a quick story, a quick example about a way that I think this could work. A way that I think is kind of that illustrates how we can find out this, the kind of known unknowns. Okay, let's do it. So there's this guy in Brooklyn that we know, and he is the head of this rare disease foundation, and families that have just crazy inherited rare diseases that hospitals and doctors can't explain go to him to try to explain the disease using genetics. And the setup is that there's this family, he told us this story, but this happens to him every few weeks. This family goes to a prestigious hospital in America, has their DNA sequenced, and they come back with, we don't know. This could be a fatal disease. We don't really know. We have these 2 DNA variants that come back that make this person unique. And there's a really, really bad phenotype. But we can't explain why these DNA variants cause this phenotype, because they haven't been seen before, right? So the variants are unclassified. That's what they're called. The diagnosis is unknown. The family doesn't know what's going on. But the hospital has done its job, right? And the normal process there is that it takes years of research to understand why this particular DNA variant causes a disease. That's how we've been, as a society, collecting information and collecting knowledge. And so this Rare Disease Foundation was a year into that process when another family with that same exact phenotype went to a different hospital. Also, they came back, okay, unknown variants, and that family went to him, and he was able to just match up on his computer these two data sets, and he saw that the variants were exactly the same. And so because there were two totally separate cases, that allowed him to explain this disease, that this variant caused this disease. So instead of years of lab research building a molecular model on zebrafish and mice, whatever, he was able to use two separate pieces of clinical evidence together. Now they explain this disease. This is huge, because all of a sudden, just by aggregating two data sets, two very small data sets, examples, with standardized evidence, you're able to make a new discovery that explains a disease. People can then screen for it later, like create drugs for it. And this happens a lot. And the take-home message is that if people can figure out how to aggregate genetic data with clinical evidence in a way that other people can use, then that will completely change the field and really explain a lot more. And so the government is engaged in something like this, and we're supportive of it. But it's just like another FTP site where people submit things. And the problem with that is that it doesn't work with everyone's understanding of privacy because it's just a public site. It doesn't work with everyone's understanding of IP, because some companies collect data, they consider it their IP, some companies don't. Different attitudes there. And also, it's just an FTP site. So people dump data in there. There's no real incentive necessarily to do so. Some companies do it for branding, sometimes publicly funded research. They do it because they feel like they should. But no one trusts other people's submissions, we found, because the standards are different. It's just an FTP site. And at least the process for submitting is easy. You just have to fill out 250 columns on a spreadsheet. So that part's fine. That part they've solved. But this is a huge opportunity for people that know how to standardize data sets, parse, munge data, and we think that kind of the data infrastructure for genomics, the platform that we've built, can be helpful. The idea is that a lot goes into going from a DNA to a clinical report. That's software. Alex has mentioned a huge part of it. There's also the ingesting data from the outside world, which is where we help, and we think that if there's a way to build in a system for people exchanging information, distributing it in a way that respects their privacy and IP, in a way where two siloed institutions or individuals can come together to form something that's together valuable, where separately isn't particularly valuable, that can really drive the field forward. And as a contrast to the FTP site, first of all, we're opening up our beta. You can sign up. Interestingly, I don't think you can access it from within Bloomberg. I think it's a conspiracy. That's the only explanation. But in contrast to the FTP site, we make it a little bit easier. You can access public data like the clinical data set I showed you with just one line of code, because we made an API to all that data. We have really great interfaces so that you could explore any DNA variant you want with all the data that's available to annotate it from the outside world. So anyway, we're better than an FTP site, is what I'm trying to say. Okay, so that's where we are. Email me, mark@solvebio.com. Follow me on Twitter or in real life if you're confident. Okay, is that? Okay, thanks.
Thank you, Mark. So the fundamental driver why people in the clinical world and other parts of the ecosystem would want to work with a platform is that it's notably better tools, better experience, because it's been very hard historically to make all these people work together. Right? It's not just a question of format.
Right, exactly. So there are a number of factors. So when it comes to ingesting data, there's one approach where everything can be built vertically. So you take all the data that's available from the outside world yourself, and maybe you make partnerships with people that provide that data. And up till now, that's in some sense been the case. 23andMe has their own curation, et cetera, et cetera. But the way we kind of see it is that inflection point has already happened. It's somewhat akin to the microprocessor that forced a lot of horizontalization, deaggregation of the industry. I think that's happening in genetics. And this is a great building to be in to talk about that, because in finance, that exact thing happened. I mean, the idea of a platform delivering data to an industry and making every player in the industry better, I think that's not too crazy when you're sitting here. So that's one of the motivations, yeah.
Great. As we're getting close on time, I want to open to questions. So what's on your roadmap, I guess? What are you?
Well, the main thing is figuring out all the right incentives in the security structure to be able to allow people to expose data to partners, but kind of have access controls so that people that they don't want seeing the data won't see it. So that's I think that will actually take a really long time. That's kind of what we'll be working on for a long time.
Great. Yes. Hold on.
What?
This is a crowd that really wants to know about money.
Yeah, I guess I'm just trying to understand what the revenue model is.
Yeah, I feel like you asked Ron that also. Yeah, so what is this, like 2006? We don't need revenue. Kidding. So we think that there's a lot of information that, and we don't think we know, there's information that people want to monetize, and they need channels to do so. So people collect data, they create their own interpretations of data, and they want to distribute it to people that use that data. So farmer wants to consume data that someone else has collected. We've seen 23andMe did an enormous deal from data that they had. The reason it was valuable is because they had all the proper consents for this fairly rare collection of variants and clinical data associated with it. That's going to happen more and more, and it's already starting to happen. And there are different kinds of data. There's clinical evidence, and as much information that you have there, the more valuable that is. And that will never go away. There's always more information that you need. And then there are just kind of interpretations on top of the genome. And also, those are things that you want to kind of distribute. It's similar in some sense to Bloomberg. There's a platform and a channel to distribute information. And if you have a good channel to distribute information, like Netflix or Bloomberg, et cetera, some of that content can be open, sometimes premium, sometimes you make your own. So I think things like that.
Great. Time for one more. This already. Okay.
Yes. So I know that from your presentation, you really made a point of the quality of the data and how the data is accessible. Mm-hmm. And knowing with ClinVar and OMIM and CBI and all these sets, how are you guys really controlling that quality? How are you doing the QC for that data?
Yeah, so the first thing that we do, the first part of that, the accessibility, and just the kind of the first level, quality level one, is we kind of re-index the data and normalize it. So as you know, everything is kind of in different formats, different indices, different versions. It could be this build of the genome, different build of the genome, missing certain fields, has extra fields for some reason, or just like a whole record's missing. So we fill all of that in. So initially it's been a somewhat manual process on our end. It's taken quite a bit of work. But the sort of next kind of layer of that is that interface that I put up where you could actually explore variants that has feedback mechanisms. And so I think one thing that analogy that I like to make is, if the human genome was quote unquote mapped in 2000, I think there's one still needs to make the Google Maps of that, and then after that we would need to make the Waze of the human genome, where you actually have feedback, where if something is correct, not correct, from a patient in a real-time way. I think that could be really promising.
Okay, well, terrific, Mark. Thank you
Cool.
very much.
All right, thanks a lot.